Welcome you all to the next class on inorganic chemistry of life principles and perspectives. Just in the previous class we have discussed a few enzymes of iron, particularly those of oxygenase type. In that we have looked at the monooxygenase and then the dioxygenase, monooxygenase based on heme, also non-heme, which is methane monooxygenase, and then based on the dioxygenase based on the non-heme, which is protocatechate. So, all of these we have looked at. I think now it is the time to move into the topic, and the iron enzymes are a little different, particularly those on the reductase properties. So, to get into this reductase properties, I would like to make some kind of a subclass of the iron proteins or iron enzymes, which are based on the binuclear iron center. Let us look at this particular table first, and I will tell you something which are familiar already and something which is not familiar already. So, we can look at those ones. So, all of these given in this are binuclear iron enzymes. Number 1, we can see that the hemerythrin. Hemerythrin we have already been aware. Where did we study the hemerythrin? I am sure if you go back and look at the oxygen transport, more detailedly we studied for the myoglobin and hemoglobin, but then we compared with that of the hemerythrin and hemocyanin because these are found in other kinds of organisms. So, therefore, we are familiar with the hemerythrin. Hemerythrin is the oxygen transport protein. Then we are also familiar recently with the methane monooxygenase, again a di-iron containing enzyme, which converts methane into methanol, of course plus water, because in monooxygenase you use only one oxygen for oxidation and the second oxygen goes as water. Now, let us look at something newer ones. The newer ones, one of them is ribonucleotide reductase. So, it is a reductase family and it does some reduction. So, what it does? It does generate the nucleotide which are the deoxy type from the ribonucleotides. So, therefore, you have a ribonucleotides to deoxyribonucleotides because to synthesize DNA you need deoxyribonucleotides, and deoxyribonucleotides are not entry to the body. Therefore, ribonucleotides have to be reduced to the deoxyribonucleotides, and more details we will discuss bit later. And so, therefore, one more enzyme in that, and then let us look at ribritrin. Ribritrin is the one which is catalyzes the breakdown of the hydrogen peroxide, and hydrogen peroxide is generated from the enzyme of superoxide dismutase. One of it is water, one of it is oxygen, other is H2O2, and the H2O2 is further reduced, further broken down to water, and you can also call it as a catalase. So, ribritrin gives a catalase kind of an activity, and this is also di-iron. We will look at the details. Then, having looked at the reductase and then the H2O2 breakage, let us look at some acid phosphatase. So, acid phosphatase is the one which basically breaks down the phosphate ester bond. So, in other words, hydrolyzes this one. And so, these three cases we are going to study a bit more detail, but in addition there are some other di-iron stearoyl ACP desaturase. It is because there is a double bond the desaturase. So, it creates a kind of a unsaturation in the saturated thing, and this is the kind of a conversion. But we are not going to look at the details of this particular enzyme because these are not so much very well known. So, having said that, these are the kinds of things, and I talked about these are all binuclear iron enzymes. Let us look at their structures, and the known ones and the unknown ones. So, we know already methane monooxygenase, as you can see here, and we also know which is the hemerythrin. These are the things, and the ones which are ribonucleotide reductase, which we are going to study now, ribritrin, which we are going to study now, purple acid phosphatase, again we are going to study now. So, and all of these, see, di-iron, all the di-irons always are connected, always are bridged by the carboxyl group. Most of the times, I would say more or less always, you take even other di-iron centers or dimetallic centers, most of the times dimetallic centers are bridged by carboxylates, and the same thing is true even the di-iron centers. You can see a carboxylate. Carboxylate comes from where? Comes from the side chain of the only two amino acids. Which are those amino acids? Which is aspartic acid and glutamic acid. The only difference is one methylene group difference between the aspartic and glutamic. Okay, so therefore, but there is an end, the side chain end carboxylic group, and which is involved. So you can see a bridging here, you can see a bridging here, carboxylic bridging carboxylic, and the only case here is bridging, but a little differently between the two, and the bridging is there and the bridging is there. So more or less we can say among the di-iron and many of the dimetallic centers, and this the two metal centers are connected by the carboxylate bridge, and the carboxylate being coming from the either from the aspartic or from the glutamic, and majority is in the case of the glutamic case. Now see the other differences. The other differences is here you have one histidine on either side, again one histidine on either side, and then in histidine and tyrosine, and two histidines, etc. And then so there is a little bit of coordination differences on all these things. What you cannot see from this slide is that the protein structural variation is also expected. So in each of these cases protein is different. The protein structure is different. That is not reflected in this slide. I would tell you that you need to take as a granted in that. And when we look at the some of the proteins, we will see that among all these, the maximum the histidines is in the hemerythrin. You have a 3 histidines and 2 histidines on the other side, and you have a 6 and 5 coordinated kind of thing where the O2 gets bound, etc., and then gets stabilized as HO2 minus species in this. And the other case, yes, so this is an important, then you have all other cases have got some histidines or the other. The ribrithin having the least number of histidines in all this. Okay, so let us try to look at some case. For example, let us start with some reductase, ribonucleotide reductase, one in more detail. Okay, ribonucleotide reductase, as I told you, and let me repeat once again, is involved in the synthesis of deoxyribonucleotides. Okay, and the body entry is ribonucleoside, and you need a deoxyribonucleoside because the cell has to synthesize the DNA, cell requires the deoxyribonucleotides. And you know cell progress and cell division, when the cell division takes place, you know the nucleus material, DNA material also gets divided. So therefore you need to synthesize the DNA. So to synthesize the DNA you need the deoxyribonucleotides, and the entry to the body is ribonucleotides. So conversion from the ribonucleotide to the deoxyribonucleotide is in that portion. One of the first and foremost is that ribose to deoxyribose, and that is converted by this enzyme. So this enzyme is very important for the cell division, cell growth, etc. So the substrates for such an enzyme are the diphosphates, adenosine diphosphate, guanidine diphosphate, cytidine diphosphate, uridine diphosphate, and some triphosphates as well. So therefore from the ribosyl to the deoxyribosyl, that we need to look at that. So in this case, if you look at the enzyme, is a bit like complicated. It has a dimeric structure, and the dimeric structure and each of it has got a unit, the RNR1. Ribonucleotide reductase is in a short form, we refer it as RNR. Do not get confused with anything else, and in this case you have a situation with 1 and RNR2 to subunit, and this 2 heterodimeric subunit, and this is twice to that. So, it is a heterodimeric tetramer you have. So, that is where the thing is. So, if you look at this further interior, let us look at for a while reaction. So, this is the ribose, you can see that, and this is the deoxyribose. So, what is the difference? This part is same, this part is same, only this part. So, 2 prime which has a hydroxyl becomes deoxy. So, dehydroxy you can say either way too, and then this is replaced by this OH is replaced by hydrogen. So, therefore, this is 2 prime deoxy ribonucleotide in this, because the base is there and the phosphate is there, it is a nucleotide. Okay. So, this is the reaction that happens, and then let us look at what is the ion center. You take the ribonucleotide reductase, as I said there are 2 parts of that is there, one is called the RNR1 part, the other one is the RNR2 part. So, therefore, and in this you have the enzyme where the reaction occurs. How the reaction occurs, that we will see in a while, and one of them is the ion center, di-iron center. So, you take a protein with the apoprotein, that means the ions are removed, and start adding the iron 2 salts, you will start seeing the iron 2 core being built in. Now, this is when it is exposed to oxygen, it will go to the iron 3 kind of a form. Of course, it is not very well understood in terms of the product, but iron 3 is very well understood because of from the EPR and other studies. So, at this stage an electron reduction will activate this part of it and will generate the tyrosine kind of a species which is involved, and that will make the oxidized form further intra redox form will lead to the tyrosinyl radical. Of course, this tyrosine radical is also stabilized by various factors which we will see in a while, and there is also a hydrophobic, there are some residues as well as a hydrophobic core which keeps this tyrosine radical intact. Let me tell you one point, never forget with this, if this radical is not formed, this is not reactive. So, this ion center is active, catalytically active only when the tyrosinyl radical is there. So, therefore, these two are basically interconnected. So, the existence of this, the catalytic phenomena of this protein is existing only when you have both of these two together. Okay. So, therefore, you are oxidizing, and when you oxidize the ion, then as mentioned, the oxo bridge will also come. Okay. So, therefore, this oxo bridge is already there, and this oxo bridge and the oxidized iron center now is sufficient enough with the electron reduction to take to a radical of the tyrosine radical. So, tyrosine radical is somewhere around within about 4 to 5 angstroms from the di-iron center, and it is stabilized by some residues, side chains, as well as a hydrophobic compartment. And as I told you, and I repeat to say that the radical and this center, oxidized center, both are essential for the catalytic activity, otherwise there is no reactivity. So, whatever I said in this, let me put in a little different format. I take an apoprotein and to that you add the iron 2 plus, and then you get into the oxygen exposed, then you get iron 3 oxo iron 3 kind of a thing. And this when in the electron reduction presence will give the tyrosine radical. So, therefore, this whole unit is very essential to that, and in the oxygen that is added, one O is used for bridging, other O will go as a water, that as you can see here, and an excellent kind of thing. So, the whole system is required. Let us look at a little bit more details. So, I have talked to you the di-iron center, and the di-iron center is stabilized by as a di-iron diferric or di-iron 3 center is stabilized with the tyrosine radical. This all thing is present in the RNR2. Okay. So, this radical is further stabilized and there is a hydrophobic core. And as you can see in the previous slide itself, then there is a lot of glutamate residues which also stabilize this one, the ion center, and that is also important. And now this RNR2 and the second part or second subunit is RNR1 as labeled, and these two are in close proximity, and the C terminal of RNR2 is in close proximity with the C terminal of the RNR1. So, with that you can understand, that means there must be some kind of a communication going on between the RNR2 with the RNR1, where the C terminal of these two proteins are interconnected or very close by. I am not don't say that interconnected, but they are close by. That means certain residues at the C terminal of RNR2 and the certain residues of the C terminal of the RNR1, they are the ones which are mediating the connectivity or mediating the enzymatic reaction. Okay. So, therefore, the enzyme activity is one thing is dependent on the stability of the radical and the dipheric center, and also is dependent on how these two units are associated. RNR1 and RNR2 are associated closely at the C terminus. Okay. So, this is absolutely essential. So, and what is there in the RNR1, there is a dithiol function, and we will see how is it important. In the RNR2, it is the diiron center, dipheric center with tyrosine radical, that is important. In the RNR1, it is the dithiol group which is equally important. How that is important, then we can look at in the next slide. So, having talked about the diiron center, tyrosine radical stabilization in the RNR2, dithiol center in the RNR1, and these two are connected through at the C terminal, and there are certain residues coming from the RNR2, the certain residues which are coming from the RNR1, there is a communication in these ones. And let us look at those which the residues are associated. In the tyrosine radical, as I said, radical is placed with certain residue side chains. They are also placed with certain the hydrophobic core, and the kind of the residues are there: aspartic 273, and this is the tryptophan 48 and tyrosine 356. So, these are from the RNR2 side. That means this radical is just not shown this the core. There is a core, the protein core, which is protecting this with a hydrophobic as well as the aspartic, tryptophan and tyrosine. And coming to the other part, which is the RNR1, there is an electron transfer, there is or radical transfer going from the RNR2 to the RNR1. This whole region is RNR1. So, here the RNR1 is going through via some residues. Here that radical transfer or electron transfer goes through the tyrosine 356 and tyrosine 731, and this continues via tyrosine 730 to the cysteine 439. So, going from one tyrosine to the other tyrosine and then the other tyrosine and then to the cysteine. So, that is how this whole thing happens. One another aspect I have forgotten to tell you in this aspect is that RNR1 is also having a compartment for binding of the substrate. So, what are the substrates here? The substrates here are the ribonucleotides. The product is deoxyribonucleotide. So, therefore, and I have shown you earlier, there are different kinds of ribonucleotides, the ADP, GDP, CDP, UDP, etc., etc. All of these are recognized and bound. All of these are recognized and bound by the RNR1. So, RNR2 has only one component, that is the dipheric ion with the radical center, and the RNR1 has got the compartment for binding to the substrate and substrate specificity. Substrate is being recognized. Anything else will not fit here. Anything comes here will not be recognized by the enzyme. So, I have told in the beginning of these courses that for an enzyme action, substrate should be recognized. So, substrate specificity is important. So, substrate specificity is also there in the ribonucleotide reductase, but in the RNR1 compartment. So, now RNR1 compartment has a substrate binding, substrate specificity and dithiol, as you can see here, dithiol, the two cysteines of these. And there are some residues in this vicinity, there are residues in this vicinity which I have not shown here for clarity. Now, let us look at the mechanism of such a kind of a enzyme. So, as I said, there is a radical at the tyrosine, and the C terminal is interfaced with the C terminal of the RNR1. And this radical is basically generates a radical at the cysteine at this interface into the RNR1. And in turn this radical is being transferred to the ribosyl compartment, because ribosyl is recognized and fixed exactly at this particular position, and it is a selective. So, therefore, the radical is transferred to the not the C2, but the C3 prime, and this radical converts by this to the position of this 2 prime, and in this process the OH is combined with the hydrogen dot, and that will leave as water. So, the water is lost, and so therefore, now the radical is being generated over at this 2 prime position, and this 2 prime position radical will pick up the hydrogen dot radical and make the C2H. So, now C2 prime has got a hydrogen, that means deoxy, and that results in the partial oxidation of the disulfhydryl functions or dicysteine, and this will transfer one more electron to this, and therefore, it becomes a disulfide. So, the partial disulfide to disulfide, and that will result in the hydrogen shift over to this, and then to the carboxylic, this carboxylic group that you can get back to that. So, therefore, you have different kinds of reactivities. So, therefore, this radical induces the radical formation at the interface, and this radical is transferred to the 3 prime, not the 2 prime, and there is a radical racemization takes place or radical transformation taking place to 2 prime, and then the 2 prime picking up the hydrogen and 2 prime H, and that is reflected in the sulfhydryl functions into the semi and to the full disulfide formation, and that leads to the hydrogen transfer over here and the radical, and this is further reduced, and this is reduced by some other the thioredoxin, NADPH thioredoxin. So, the thioredoxin is another enzyme, that enzyme gets and brings back to the normal. Okay, and this is how it is. So, what are the events that we have learned in this ribonucleotide reductase? Ribosyl to the deoxyribosyl is absolutely essential for DNA synthesis. So, all that you need is what do you need? You need to remove this OH and put hydrogen, that is all you need. So, that is going through the following mechanism because of this. So, there is the reaction, though this is the diion center with the tyrosine radical is important, direct reaction does not directly occur at this, as you can see, because the substrate is bound away from it, not in the RNR2 compartment, but in the RNR1 compartment. So, substrates binding, substrate recognition. This is one of the cases where you have a bit of deviation, that direct substrate binding at the catalytic center is not happening. So, here we can call as a catalytic center or inducing center, and that will induce the radical into this particular sulfhydryl function cysteine, which is transferred to the C3 prime, converted to the C2 prime, and then pick up a hydrogen and the release of this to form this SS semi oxidized to the fully oxidized form. We will get back to that, and this disulfide which is formed is further reduced back to the sulfhydryl by the thioredoxin process. See, very nice kind of a ribonucleotide reductase kind of thing. So, ribose to deoxyribonucleotide. So, we understand there are two parts, ribose 1 RNR1 containing the ferric ion tyrosine radical, and the RNR2 containing the diferric ion center with tyrosine radical, RNR1 containing the compartment for the substrate binding as well as the sulfhydryl function. So, we have seen the total role of this enzyme, the two compartments. Having seen this, let us move to the next enzyme of the diiron, which is the ribritrin. As I mentioned already, it catalyzes the breakdown of the H2O2 to water. So, that means it is a catalase activity. It is essentially a catalase kind of an activity. So, you can see again this is the kind of an enzyme structure with a huge alpha helical structures, the two interfaces, and this is how the interface is connected, head to tail dimer interface. So, the center difference between this, distance between this is about 12 angstroms. So, whatever the electron transfer takes place, it takes place through this kind of thing. So, this reaction is a very well known reaction to us by now. And how does this happen? This is the diiron center, and the diiron center, when the substrate, here the substrate is H2O2 and then H2O2 binds. This is not proven species, and so therefore, that reaction goes from the redox process that is occurring at the ion centers, and that cleaves and loses one water and forms a oxo bridge. Whenever the ion 2 goes into the ion 3, both the ions go into ion 3, always the oxo bridge that we have seen that. And this is further reduced by the ion sulfur protein called the rubridoxin. So, that is why it is called ribritrin. Rubridoxin will reduce back this one, and the protonation also takes place. So, at this stage one more oxygen is reduced to water, and that will go back, and again rubridoxin further reduces and brings back to this. So, the diiron center. How do we know this is the really center that is responsible for the reactivity? Now, you react with the HN3. So, the HN3 will bind in this region where otherwise the H2O2 binds. So, H2O2 is binding here, and the same place HN3 will bind, N3 minus will bind. So, when the HN3 minus will bind, this will inhibit the reaction. So, this clearly tells this is the reaction center. Okay. So, now we have a diiron center, the H2O2 binding leading to the oxidation of this, breakage of the H2O2, one water and one oxo bridge, further reduction by the rubridoxin and reducing the ion center, further reduction leading to the protonation, leading to the one more water generation, and that will lead to the starting part of that enzyme. So, we know this is the region where diiron center is the one where the hydrogen peroxide is bound, and this is being shown by this particular azide binding, N3 minus binding. So, what we learn in all this is whenever you have the diiron centers, particularly when the two ions are 2 plus and going from 2 plus to the 3 plus from a reduced protein called diiron 2 and oxidized protein called diiron 3, whenever you go from reduced to the oxidized protein, you get an oxo bridged. This can be coming even from O2 breakage mostly. And then the other one will go in the water, and this is where you can see this is for the oxidized, the ribonucleotide reductase, reduced ribonucleotide reductase, you can see. Similarly, here the O bridge extra here. Then in this case methane monooxygenase, the oxidized, there is oxygen, then that oxo bridge is lost over there. So, you can see in all these diiron centers what you have is essentially is that the two ion 2s which are in reduced form will go to the oxidized form, and that will pick up an O and then oxo bridged. So, what we have learned, looked at the two enzymes. One is ribonucleotide reductase, which converts the ribonucleotides into deoxyribonucleotides, and the second one is H2O2 catalase breakdown of this. So, in one case the diiron center is not a directly catalytic center because the substrate binds somewhere else, but it induces what is required. What is required? The cysteine radical, and this radical further transforms as I explained to you, and then finally this converts this 2' OH into 2' H, and I have told you all the steps going through that. And there is a dithiol in the RNR1 which undergoes oxidation, and then at the end it will be reduction. Analogous, and in the case of the ribritrin, the H2O2 is the substrate, is directly bonded to that. How do we know? Because the azide N3 minus binds here and inhibits the reaction. So, we have looked at that too. So, both the di-iron enzymes, when they are reduced, they have oxo bridge is not there. When they are oxidized, their oxo bridge is there. I hope you keep these things in mind, and we will look at in the next class on something on the phosphatases kind of thing. In other words, some kind of a phosphate bond hydrolysis of this. Thank you very much.